Hi everyone. The practical this week is entitled Dilutions and Standards, otherwise called the Tartrazine Standard Curve. So our learning outcomes this week will be to be able to make a stock solution from a solid compound, to make a series of dilutions from that stock solution, to construct a standard curve from the dilutions that you've made, and then to calculate the concentration of an unknown that you will be given from this standard curve. So first of all, when we're preparing solutions, we can prepare either a molar solution or a mass solution, a mass concentration. So the molar solution is defined in moles per litre, and we discussed these during the lab calculation session, and also a mass solution, which is essentially a particular mass per volume, so grams per litre, for example. For molar solutions, you need to know the molecular weight of the compound that you're using. And for this, we're using distilled water to make up solutions and to stir. So stock solutions should always be made up in a volumetric flask. This is because it has a very precise and exact volume attributed to it, and it's calibrated. So always make up a stock solution from within a volumetric flask. For this week's practical, we're going to be using a substance called tartrazine. Tartrazine is just a simple food colouring. So it's otherwise known as E102. And it's water soluble, and it has a maximum absorbance of 427 nanometers. And its molecular weight is 534.4 grams per mole. This is just showing you here in the diagram a picture of its chemical structure. So to make the stock solution in this practical, you're going to be asked to make a 0.1% WV solution of stock solution of tartrazine. So if you remember back to our lab calculation session, 0.1% means 0.1 grams per 100 mils, not per litre. It's percent, it's out of 100, so it's 0.1 grams in 100 mils of distilled water. So as I said, this has to be made up in a volumetric flask in order for it to be accurate. And to do that, you first of all need to dissolve it in a reduced volume of water and stir it. So in this case, we're getting you to weigh out 0.1 grams on a weighing balance. So don't forget to zero the balance. Weigh out the powder onto the weigh boat, and then you transfer it to a container with water, roughly about 70-80 mils of water. And in the container, you use a stirrer, a magnetic stirrer, and you stir it for a couple of minutes. Then using a funnel, you pour it into the volumetric flask. You then will have probably some distinctive orange color at the bottom of your container. If you wash that out with your distilled water bottle, and add the washings then again via the funnel to the volumetric flask. Finally, once all of the orange color is gone, you've removed all the water from your container, you can then add dropwise to the volumetric flask using something like a pipette up to the meniscus. So the meniscus is the point here on the diagram, the line on the volumetric flask, and you add it dropwise until you get this nice U shape onto the meniscus. This is known as the volume graduation mark. Okay, at that stage, then you put the lid on and you invert it a number of times to make sure that it's well mixed. So once you've the stock solution made, you then have to prepare a series of working dilutions from this stock solution. So this is what we're going to use then to make our standard curve. So these will be solutions of different concentration. So we're going to dilute the stock solution at the beginning, and at the end we'll have 1,000 microliters of each standard then within like an Eppendorf tube, so a 1.5 mil Eppendorf tube. And then from that we're going to pipette 100 microliters of those dilutions, of those standards, into 96 well plate several times. So we're going to pipette them 4 times for each different standard, so in quadruplicate. In addition to that as well, you're going to be given a Eppendorf tube containing an unknown concentration of tartrazine. So we don't know what's the concentration, and we're going to use then the standard solutions that you've just made to estimate the unknown concentration. And we do this by reading the absorbance on 450 nanometers on the spectrophotometer or plate reader. So similar to last week's practical, where you had to measure the absorbance using the food dye, you're going to read it at 450 nanometers, get a printout from the spectrophotometer, and use it then to construct the standard curve. So you'll have to get the means, standard deviations, and the percentage variance for each of your standards and your samples, and also then for your unknown. Using the standard means, you're going to plot a standard curve by plotting the concentration in micrograms per mil on the x-axis versus the absorbance, which you've just read off from the spectrophotometer, which is arbitrary units, on the y-axis. And then you're going to determine the concentration of the unknown by extrapolating the absorbance of that unknown and moving across from the y-axis where it meets the straight line, the standard curve, and dropping it down then to the x-axis to estimate the unknown concentration. So you've been given this table in your lab manual, and you see the concentrations that are required. So we have our stock solution, remember, at the beginning in the volumetric flask, and then we have a series of unknown of standard concentrations of 100, 80, 60, 40, 20, and then finally 0. And the table then asks you to work out what dilution of that stock solution would you need in order to get this concentration of 100 micrograms. So you need to work that out first of all, and then you need to work out the same for each of these other values. So it requires you to estimate the number of microliters of the stock solutions taken straight from the volumetric flask, plus water, in order to give you this final concentration. For the other wells, you'll get a background absorbance. These have to be, and particularly for the 0 here, you'll have to subtract the absorbance for the 0 from each of these other values here, okay, including your sample as well, your unknown, before you can plot them on the graph. Okay, so once you've plotted the graph, you can then extrapolate your unknown value, both the neat, the 1 and 2 dilution, and the 1 and 5 dilution, in order to get an estimate of the concentration. And don't forget, at the end then, when you've done the 1 and 2 and the 1 and 5, you need to remember to multiply your answer by 2 and to multiply your answer by 5 in order to give you a correct estimate. You then average the 3 values that you get, so for the neat, the 1 and 2, and the 1 and 5, to give you your final answer. Okay.